Now, good morning to all of you. This is the second part of the principles of inheritance and variation. In the first part, we have studied about the simple introduction of the Mendelian genetics as well as the inheritance and variation. In this part, we will study about the Mendel's law of genetics or Mendel's law of inheritance along with the certain genetic terminologies. So now we start the topic. Here, Mendel has selected 7 characters of the pea plant for his experiment. Why he has selected pea plant? Actually, pea plant is annual plant. Its lifespan is of 2 to 3 months. It means in 1 year we can produce 3 to 4 crops of the pea plant. The flowers of pea plant are well exposed, easy to study. Its male and female part are well distinct. So we can easily study about the floral structure and sexual structure of the pea plant. Since pea plant has certain distinct and observable characters that can be studied in different generations, so Mendel has selected the pea plant for his experiment. Pea plant can be cultivated in any type of soil at everywhere. This is one of the reasons behind the selection of pea plant for this experiment by the Mendel. So Mendel has selected 7 characteristic features of the pea plant for the experiment, for the study. Now these 7 characters are seed coat color, seed shape, seed color, pod color, flower position, pod shape and stem length. So these are the 7 characters. Each and every character is shown in 2 forms. So let's see: coat color gray and white, seed shape round and wrinkled, seed color yellow or green, pod color green or yellow, flower position axial or terminal, pod shape inflated or constricted, and stem length or plant height tall or dark. So these are the 7 characters. Here these are the 7 characters: seed coat color. Seed coat color and flower color, these 2 are taken as single character. In the seed coat color, there are 2 types of seed coat or flower colors are shown: grey and purple. It means here the color grey is also color, purple is also color, and white. So seed coat color or flower color may be of 2 categories, either color or white. Seed color means inside the seed coat, the color of pot leader is considered as the seed color. So the yellow and green, this is the categories of seed color. So the yellow and green. Now seed shape, seed may be round shaped or wrinkled. Pod color green and yellow. Pod shape inflated and pinched or constricted. In intricate pod shape, we can't say about the number of seeds present inside the pod without opening the pod. But in the constricted pod, we can say the number of seeds present inside the pod without its opening. Now, next is the stem height or height of the plant. This may be tall and short. And the flower position. Here the flower position as well as pod position. Both are taken as single character. Flower position or pod position may be axial or terminal. Here this is the axial position and this position is called as terminal position. Now this is the terminal position and this position is called as axial position. So the terminal position is this. If the flower or pod is at the top of the branch, then it will be the terminal, and if the flower or pod is present at the axillary position, then it will be axial. So these are the 7 characters, and all these 7 have 2 optional or 2 behavioral conditions. In the seed pod color, this may be colored or white. In the seed color, yellow and green. In the seed shape, wrinkled and smooth. In the pod color, green and yellow. In the pod shape, inflated and constricted. In the stem height, tall and dark. And the flower position, axial and terminal. So these are the different characters of the pea. Here, Mendel has collected the data of these 7 pea traits after the experiment done by Mendel. Here, there are 2 types of the characters or behavioral characters or traits expressed by the pea plant in its lifespan. One character is called as recessive. One form is called as dominant form, while another is called as recessive form. What characters expressed by the plants are considered as dominant? Now they are the dominant characters and these are the recessive characters. When we study about the color of flower, then the color of flower or purple flower is the dominant character, while the white flower is the recessive character. It means the dominant trait is the color of the flower, while the white flowering or color of flower is white is the recessive form of the pea plant. It means when we study about the dominant character, it means the dominant character is that character which shows or expresses itself in the next generation. That will be called as the dominant character. And recessive characters, the characters that cannot be expressed in the next generation whenever it is in the heterozygous condition. What is homozygous? What is heterozygous? We study further. Now, when the dominant form and recessive forms are crossed together, the ratio of dominant and recessive forms in the F2 generation was 3.15:1. While the yellow seed, that is the color of seed, means color of cotyledon, it is the dominant. Green seeds, recessive character, and the ratio was 3.01:1. Round seeds and wrinkled seeds, roundness was the dominant character and wrinkledness was the recessive character. And now the ratio was 2.96:1. Green pod and yellow pod. Green pod, possession of green pod is the dominant character, possession of yellow pod is the recessive character, and now the ratio was 2.82:1. Inflated pods and constricted pods. Inflated pods are the dominant character, constricted pods are the recessive characters. Now the ratio was 2.95:1. Axial flowers and terminal flowers. Axial flowers, the presence of flowers at the axial position is the dominant character, while at the terminal position is the recessive character. Now the ratio was 3.14:1. And the last, tall plants and dark plants, the ratio was 2.84:1. When we make the figure round up, then it can be considered as 3 is to 1, 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 or 3 is to 1. Now, what did Mendel find? He discovered different laws and rules that explain factors affecting heredity. On the basis of the experiment, inferences and conclusions from his experiment, he has given the laws and rules of heredity or inheritance. That laws are considered as laws of Mendel's, or Mendel's law of inheritance, or Mendel's law of genetics. Now, here the rule of unit factor. What is the rule of unit factors? Each organism has 2 alleles for each trait. What are alleles? Alleles, the different forms of the same gene. The different forms of the same gene. And genes located on chromosomes, they control how an organism develops. Actually, at the time of Mendel, gene term was not coined. So alleles means for each and every trait has 2 forms, and these 2 forms are characterized by the alleles. Suppose that the tallness and the dwarfness. The tallness and the dwarfness are characterized by the 2 types of alleles. Now, the trait that is observed in the offspring is the dominant trait, and it is written in uppercase or capital form. The trait that appears in the offspring, the trait that disappears in the offspring is the recessive trait and written in the lowercase or small letters. Here capital T that denotes the tallness of the plant or height of the stem. Capital T means it represents the stem, the height of a stem is longer or tall plant, while the small t is for the dark plant. When plants having one pair of character, tall and short plant, was crossed, that is considered as a monohybrid cross. Actually, it is another term, crossing. When the breeding is being done between the 2 characters, between the 2 individuals, and focusing on the single character only, then it will be considered as the monohybrid cross. Mendel selected pea plants having one pair of character, a tall pea plant and a short pea plant. He selected pure tall and pure short pea plants and cross-pollinated them. He obtained all tall plants. How? Here, these are the tall and short plants. When the tall and short plants are crossed together, then in the F1 generation, what is F1 generation? The first filial generation. By filial we mean related to son, related to offspring. So in the F1 generation, all the plants were tall plant. And when the tall plants are self-pollinated or self-fertilized, then in the F2 generation, 3 tall plants and 1 short plants or 1 dark plants were produced. Here, this is another example. The color of flower, purple and white. In the F1 generation, all flowers were purple. But after the self-fertilization, in the F2 generation, 3 flowers were purple while 1 flower was white. Here are the round and wrinkled colors, shapes of the seeds. In the first generation, round seeds were developed, but in the second generation or F2 generation, after the self-pollination or self-fertilization, 3 plants were round while 1 plant was the wrinkled. This is the ratio, 3:1 ratio. This is the monohybrid cross, and it is this monohybrid cross is the base of law of dominance. Means when the two homozygous plants are crossed together. Homozygous means purebred. It means both the alleles of a particular character are similar together. It means, what is this? It means if we take the height of the plant, then capital T and capital P, both are the similar alleles. So it is called as homozygous. And if we take capital T and a small t, here both the alleles are not similar together. So this condition is called as heterozygous. And when we take a small t and a small t, again it is called as homozygous, because both the alleles are similar together. So this condition and this condition are called as pure breed, while this condition is not called as the pure breed, it is a hybrid breed. So Mendel has crossed the two homozygous plants, this and this, and after the crossing, capital T, small t, after the crossing, the gamete will be formed before the crossing, and now this gamete will be this. And this gamete are fused together to produce capital T, small t in the zygote. So now here in the F1 generation, the alleles are different together, different from each other, but the capital T allele expresses itself while a small t cannot be able to express itself. So capital T is called as dominant allele or dominant factor, and a small t is called as recessive allele or recessive factor. It means in the heterozygous condition, the trait or allele that is able to express itself is considered as the dominant character or dominant allele or dominant factor. And if the allele or character or factor is not able to express itself in the heterozygous condition, then it will be considered as the recessive character or recessive allele or recessive factor. So this first monohybrid cross, or this is called as law of dominance. This law is called as law of dominance, and it is based on the monohybrid cross, in which only single character is considered for the experiment. So I think you will be able to understand the law of dominance or monohybrid cross. In the second condition, that is called as law of segregation, Mendel's law of segregation. This law is also called as law of purity of gamete. Actually, the Mendel's law of segregation or law of purity of gamete is also considered as universal law of genetics, because it has no any exception. It is implemented for each and every organism. So this is called as Mendel's law of segregation. What is law of segregation? The two alleles for a trait must separate when gametes are formed. A parent randomly passes only one allele for each trait to each offspring. Previously I have explained that the gametes are formed. Suppose that here the round shape of seed means it has capital R alleles either, or capital R small r alleles. Here it is homozygous and it is heterozygous. Now the two alleles for a must separate when gametes are formed. When the gametes are formed or during gametogenesis, we know at this time, we know that the homologous chromosomes are getting separated. Homologous pair of chromosomes are getting separated during the meiosis, after the metaphase stage. On the basis of that concept, we can say that during the gamete formation or during the gametogenesis, this R and this R will be separated from each other. So, in one gamete, only single R will be born, while in the another gamete, another single R will be born. Similarly, capital R and small r. So, the alleles are segregating. So, the alleles are segregating during the gametogenesis. And now we know gametogenesis is the first step of the reproduction. So, the gametes are segregated or gametes are separated from each other. That's why this is the law of segregation. A parent randomly passes only one allele for each trait to each offspring. Suppose that when the fertilization is taking place, this R may be fused with this R to produce capital R, capital R. And now it may be fused with this small r to produce capital R, small r. So, which R is fused with which R is a matter of chance. That's why you can say that the parent randomly passes only one allele for each trait to each offspring. This is the law of segregation. In the law of segregation, now the pair of alleles of each parent separate and only one allele passes from each parent onto an offspring, studied earlier. Which allele in a parent's pair of alleles is inherited is a matter of chance, and segregation of alleles occurs during the process of gamete formation, during the meiosis, and this is randomly unite at fertilization. Now, these are the basis of variations also. In the introductory video, we have studied about the variation. Now, law of segregation is the basis of variation also. Here, again the same example. In the parent generation, purple and white color are cross fertilized. In the F1 generation, all plants having the purple flowers. While in the F1 generation, when we self fertilize the plants, in the F2 generation, the plants are produced of two types, in which the ratio of flowering were 3:1, where the 3 plants having the purple flowers, while 1 plant has the white flower. So, this is the law of segregation. Now, what is the difference between the law of dominance and the law of segregation? When we take only this step, then this will be the law of dominance, and when we take these all, then it will be considered as the law of segregation. It means we can say that law of dominance and law of segregation both are based on the monohybrid cross. Now, what is phenotype and genotype? Phenotype: the way an organism looks, red hair or brown hair. What is it? Actually, the observable characters are called as phenotypes. It means the person having black hair. It means the presence of black hair is the phenotype. The person having blue eyes, it means presence of blue eye is the phenotype. And what is genotype? The genetic combination of an organism. At the time of Mendel, gene was not discovered. So we can say that the allelic combination. We can say the allelic combination. But at this time, currently we know about the gene, so we can say the gene combination. So the genetic combination of an organism or the genetic combination for a particular trait is considered as the genotype. It means if tall plant and dark plant, here the tallness is the phenotype, darkness is the phenotype, but the capital T or capital T small t, these two combination may be formed for the tall plant. These two are the genotype, and for the dark plant, a small t, this will be the genotype. So the genetic combination behind the expression of a particular phenotype is called as genotype, and the character that is observable is called as phenotype. Dihybrid versus monohybrid. Monohybrid, abhi we have studied about the monohybrid cross, in which we are focusing on single trait only. But when we are focusing on more than one trait or two traits, when we are focusing on the two traits or crossing the parents along with the focusing on two traits, then it will be called as dihybrid cross. It means, suppose that green and brown seeds or yellow and brown seeds, green and wrinkled seeds or yellow and wrinkled seeds. Here is the color of seed as well as the shape of seed. These are the two characters. So when we are focusing on the two characters for the crossing between the parents, then this type of cross is called as dihybrid cross. It may be trihybrid, polyhybrid cross. If the focusing characters are more than two, then it will be called as polyhybrid cross. So dihybrid cross and monohybrid cross. Heterozygous and homozygous, already we have studied. Law of independent assortment, this will be the topic of our next video. In the next session, we will study the law of independent assortment that is based on the dihybrid cross. So, it is over for this session. Thank you.